Hi everyone, I am Sundar, welcoming everyone to my YouTube channel Master of Biology. Today the lesson is for class 12, chapter 6, Molecular Basis of Inheritance. Class 12, chapter 6, Molecular Basis of Inheritance. Under that, the DNA, deoxyribonucleic acid. So what is that? What is DNA? It is a long polymer of deoxyribonucleotides. It is a long polymer of deoxyribonucleotides. And the length of the DNA is based on the number of nucleotides present in it. The length of the DNA is based on the number of nucleotides present in it. And the chemical structure of this polynucleotide chain, the DNA, the chemical structure of the polynucleotide chain, the DNA, comprises three components. They are nitrogenous base, a pentose sugar, and a phosphate group. Again I repeat, three components are nitrogenous base, pentose sugar, and phosphate group. So let us see that one by one. So let us see nitrogenous base. Nitrogenous base has two types, purines, pyrimidines. So purines, pyrimidines. Purines include adenine, guanine. Purines include adenine, guanine. Pyrimidines include cytosine, thymine, uracil. Cytosine, thymine, uracil. In case of RNA, instead of thymine, uracil will be present. In case of RNA, instead of thymine, uracil will be present, right? So that is one change in RNA, whereas DNA contains thymine. Okay, so the nitrogenous base of two types, purines and pyrimidines. Purines include adenine, guanine. Pyrimidines include cytosine, thymine, uracil. Okay, this nitrogenous base is linked to pentose sugar through N-glycosidic linkage. It is linked to the pentose sugar through a linkage called N-glycosidic linkage to form nucleoside. That means the adenine, after this linkage, will be called as adenosine. Guanine will be called as guanosine. Okay, so it becomes a nucleoside. So let us see the next two components. Okay, next comes phosphate group. Okay, phosphate group is linked by phosphoester linkage. Phosphate group is linked by phosphoester linkage. The two nucleotides are thus linked through 3'5' phosphodiester linkage to form a dinucleotide. The two nucleotides are thus linked through 3'5' phosphodiester linkage to form a dinucleotide. The thing is, when you see the DNA structure, it's a double stranded structure. Okay, so nucleotide, when it is just two in number, it's called a dinucleotide. And more such nucleotides join to form a long polymer, it becomes a polynucleotide chain. And the DNA is a double stranded structure. At one end it will be 3' and the other end will be 5'. Similarly, vice versa. It's called the antipolarity of the DNA structure, antipolarity. One end will be 3', the next will be 5', and vice versa. So that's what they are mentioning here. So 3', 5'. Okay, I'll tell you more about this. Okay, so the two nucleotides are thus linked by 3', 5' phosphodiester linkage to form dinucleotide. As I told you, more nucleotides join to form a polynucleotide chain. More nucleotides join to form a polynucleotide chain. So what is the backbone of this DNA structure? Means it is sugars and phosphates. It is the sugars and the phosphates that form the backbone of the DNA. See, the backbone of the chain, the backbone of the polynucleotide chain is made up of sugars and phosphates. Okay. Okay. So who first identified DNA? It is Frederick Meischer. Frederick Meischer in the year 1869. He identified an acidic substance and named it as nuclein. Okay, not DNA, but he named it as nuclein. Frederick Meischer, 1869, identified acidic substance and he named it as nuclein. Okay, and you know very well that at that time there was lack of technology. Okay, there was no advancement in technology. So after many years, Watson and Crick, scientists by name Watson and Crick, they proposed a DNA model. It's called double helix model, a double stranded model. Okay, in 1953, after so many years, okay, Watson and Crick proposed the double helix model of DNA in 1953 based on the X-ray diffraction method or data proposed by Wilkins and Franklin. So Watson and Crick proposed the double helix model based on the X-ray diffraction method given by Wilkins and Franklin. Okay. Then one more scientist by name Erwin Chargaff. Erwin Chargaff, he proposed the base pair rule. Base pair rule says that adenine will always pair with thymine. Similarly, guanine will always pair with cytosine. That means a purine with pyrimidine and vice versa. Okay, a purine with pyrimidine and vice versa. Adenine, thymine, guanine, cytosine. Okay, so this base pair rule says that this kind of arrangement is constant and equals one another. It is constant. So this rule was given by Erwin Chargaff, known as the base pair rule. So let us see the salient features of DNA. So first one, DNA is made up of two polynucleotide chains. DNA is made up of two polynucleotide chains. Backbone of DNA is made up of sugars and phosphates. Sugars and phosphates. Third point, the two chains exhibit antipolarity. That is 3' to 5', 5' to 3'. One side will be a phosphate group, the other side will be a hydroxyl OH group. Okay, so 3' and 5' ends. Next comes the base pairing rule. Okay, that says purines comes opposite to pyrimidines. Purines comes opposite to pyrimidines. Adenine will always pair with thymine. Guanine will always pair with cytosine. Between adenine and thymine, it is 2 hydrogen bonds. Between guanine and cytosine, it is 3 hydrogen bonds. And base pairing offers a unique property called complementarity. Complementarity. Okay. That means if there is one strand, the other strand can be predicted. It can be predicted. One strand acts as a template. And finally, two daughter DNA can be formed. Two daughter DNA can be formed. Base pairing offers a unique property called complementarity. Second thing, purines always comes opposite to pyrimidines. ATGC. Adenine is purine, thymine is pyrimidine. Similarly, this guanine always pair with cytosine. Between A and T, 2 hydrogen bonds. Between G and C, there is 3 hydrogen bonds. Wait. So what I have drawn is a double-stranded DNA. You can see the nucleotides. It's a double-stranded DNA. So if I close with this, it is predictable. If one strand is there, it acts as a template for the other strand. A means the next will be T, G means C, likewise. So this is called the complementarity feature. So one strand will act as a template to form the other one. Okay. Okay, the other salient features. Two strands are coiled in a right-handed fashion. The way it is being twisted is in a right-handed fashion. Okay. Pitch of the helix is 3.4 nanometers. So I have drawn here, that is one helix, that is about 3.4 nanometers. And between that one turn, there are about 10 base pairs in a turn. And the hydrogen bonds present between adenine, thymine, guanine, cytosine, these hydrogen bonds provide stability to the helical structure. It offers stability. Again I repeat, strands are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometers, you can see here. Roughly about 10 base pairs in each turn, and hydrogen bonds offer stability to the helical structure. So in this lesson I have explained in detail about the DNA structure. In my video I will support my explanation with some diagrams. You can go through that. Okay. So hope you like this video. Kindly subscribe, share and support. Thank you.